Hello, welcome to this presentation, Fourier analysis of cerebral metabolism of glucose, gender differences in mechanisms of color processing in the ventral and dorsal streams in mice. This is Professor Dr. Philip Njemanze. This work was done with Matthias Kranz and Professor Peter Brust. Our affiliations are Chidika Medical Center, Owerri, Nigeria, the Helmholtz Centrum, Dresden, in Leipzig University, and both authors are from the same institution. My citation is given in this slide too. We can see the arteries of the eye. It's called, the main artery is the central retinal artery that goes directly to the eyeball and is supplied from the ophthalmic artery, which is a branch that comes from the main arteries of the brain, of the circle of Willis. In this cross-section of the eye, we can see light falls on the retina, and there are three groups of cones: the blue cone, the red cone, and the green cone, and the rods, which are for night vision. We have three classes of receptors at the retina: the short wavelength, that's the blue, which is about 435 to 450. Then the medium wavelength, and then the long wavelength in the red zone. The green zone is the medium wavelength, and you could see that in this display. Light gets in through the retina, through the left and right visual fields, and conducts through the optic nerve, crossing at the chiasma, and gets down through the optic radiation to the cerebral cortex, into the visual cortex area. Light gets into the retina and through the dorsal lateral geniculate nucleus in the thalamus, gets through to the primary visual cortex and divides into two pathways: the dorsal stream and the ventral stream. The dorsal stream gets to the posterior parietal cortex, and the ventral stream into the inferior temporal cortex. The lateral geniculate nucleus has 6 layers, and so also the steroid cortex has 6 layers. The ventral stream has the function of what? Looks at form, color perception, recognition, and object background, while the dorsal stream looks at where? Eye-hand coordination, orientation in space, according to conventional knowledge. We can dictate activity in the primary visual cortex using functional magnetic resonance imaging in humans. With color perception, we are more concerned with the fusiform gyrus. You can see located below on this slide. Visual processing takes place in ocular dominance columns for the left eye and for the right eye, and these are arranged in blobs, as you can see. The blood supply to the visual cortex comes from the posterior cerebral artery and the middle cerebral artery, mainly. The middle cerebral artery has two types of terminal branches: the ganglionic or lenticulo-striatal arteries, which diffuse the dorsal aspect of the brain, and the cortical arteries, which diffuse the ventral part of the brain. The demarcation of the perfusion distribution of the middle cerebral artery shows the deep arteries in the middle part or the dorsal part, and the cortical arteries in the ventral part. The superior parietal aspects of the optic radiation receive blood from the middle cerebral artery, whereas the inferior aspects receive blood from the posterior cerebral artery. The macular cortical region also receives blood from both arteries. The calcarine artery supplies the macular region, and then the branch of the middle cerebral artery, which is supplies this area too. That's why there is macular sparing in an eye lesion. Color processing is a brain computation, and we can illustrate this with simultaneous color contrast, where the surrounding colors influence the perceived color. And you can see in the test given on this slide, we had earlier suggested that in men there is wavelength differencing during simultaneous color contrast, and in females there is frequency differencing, and this led to further investigation which will be carried out in this study. Another quality is color constancy. Color constancy is that the color remains constant despite the light illuminating it, as demonstrated on this slide. Color blindness involving the brain is termed cerebral achromatopsia. It usually involves ischemia or infarction of the ventral occipitotemporal cortex in humans. If it is on one side of the brain, it's called hemiachromatopsia. The commonly used clinical diagnostic tests for color vision are the Ishihara plate test and the Farnsworth-Munsell 100-hue test. In 2010, we studied cerebral blood flow velocity changes during color stimulation using transcranial Doppler. We demonstrated that in men, the changes occurred in the right hemisphere and showed wavelength differencing. In women, the changes occurred in the left hemisphere and showed frequency differencing. We applied Fourier analysis. Using spectral density curves, we demonstrated that the changes in men showed right subcortical-cortical changes during color processing, but left cortico-subcortical changes in women. This is demonstrated in the spectral density plots on the right-hand side. Red for male changes and blue for female changes. In 2017, we developed a mouse model, and the mouse model demonstrated that in male mice, the changes occurred in the left visual cortex during blue light stimulation, but in the right visual cortex during blue light stimulation. There was right eye ocular dominance in male mice, but left eye ocular dominance in female mice. We then postulated that there was a major gender differential in color processing, and that the changes in male mice would occur in the cortico-subcortical ventral stream, while changes in female mice would occur in the subcortico-cortical dorsal stream, and that we could apply Fourier analysis to differentiate the changes in the subcortical area from those in the cortical region. We studied 5 male mice and 5 female mice. They were placed in a ventilated temperature-controlled cabinet with 12 hours of daylight or light and darkness cycle. They had unlimited access to water and food. The animals were studied repeatedly on consecutive days without randomization, and we kept the daytime study at the same time. There was no significant change in weight of the animals, and we carried out the test with monoocular occlusion. The experimental protocol started with isoflurane anesthesia. After 5 minutes, we injected fluorodeoxyglucose 18 and started 20 minutes of eye stimulation. Then we positioned the mouse in the scanner and started the PET scan after 2 minutes for a period of 20 minutes. After 20 minutes, we conducted a 10 minutes post-scan MRI. The experimental setup is hereby demonstrated. We used Ratin filters of deep blue and deep yellow, and the setup of the chromatoscope is demonstrated in this schematic diagram. There were 7 different stimulation conditions: dark in both eyes, light left eye, right eye, blue left eye, right eye, yellow left eye, right eye. This stimulation was conducted through a double barrel eyepiece. The PET scan was performed using a nanoscan PET MRI from Mediso Medical Systems, Budapest, Hungary. We observed the changes from the PET MRI console. The regional FDG uptake was calculated using the standardized uptake value SUV, which is the ratio of the tissue activity concentration and the injected radiotracer amount divided by the body weight. There were 4 mid-frame time points: 29.5, 34.5, 39.5, and 44.5 minutes after the FDG injection. We defined the volume of interest in the right and left visual cortex First, the volume of interest included the whole cortex. Then we drew two smaller volume of interest areas, one to the right in the right visual cortex and one to the left in the left visual cortex. We now calculated the SUV within these regions. We demonstrate the PET images in male and female mice in the right and left visual cortex. Then, in the MRI images below, and the contrast CT angiography, which shows the ganglionic branches and the cortical branches. And a schematic diagram is shown of the circle of Willis in mice and the tracer accumulation. We performed statistical calculations to evaluate the stimulus effect with paired t-tests, ANOVA and MANOVA with repeated measures. The alpha level was set at 0.05. The stationarity assumption was performed with the augmented Dickey-Fowler test using the Starter software package. We then performed the Fourier analysis using a linear multiple regression model where the dependent variable is the observed time series and the independent variables are the sine functions of all possible frequencies. We then plotted the spectral density estimates, which showed the frequency regions with the highest estimates, and they were marked as peaks. The cortical C peak, which occurred at frequency of 0.2, and the subcortical S peak, which occurred at a frequency of 0.4. We analyzed the SUV values for main effects. There was a main effect of stimulation, there was a main effect of visual cortex, and there was a main effect of gender. There was a stimulation times gender interaction and a stimulation times visual cortex interaction. The analysis of SUV values did not distinguish changes in the dorsal and ventral streams. We plotted the spectral density estimates in male and female mice, which can be visualized on this slide. We performed the analysis of the spectral data, which demonstrated that in male mice, there was luminance opponency in the subcortical region of the left visual cortex in the ventral stream, while chromatic opponency was present in the cortical region of the left visual cortex in the dorsal stream. In female mice, there was no significant response to white light. The response to blue light stimulation was frequency modulated and induced resonance phenomenon in the cortical region in the dorsal stream of the right visual cortex, while chromatic opponency was implemented in the subcortical region in the ventral stream in the right visual cortex. We then examined the cross-correlation factor. In males, Blue caused negative correlation at -3 lag. In females, blue caused positive correlation at -5 and 0 lags, and negative correlation at +2 lag. We then examined the cross amplitude, phase, gain, and coherency in male and female mice. In male mice, the coherency increased with spectral narrowing at the lower frequency range. In female mice, coherency increased with spectral broadening across the lower and higher frequency range. For color processing, there appears to exist two models, a male model through the ventral stream and a female model through the dorsal stream. The male model begins from the retinal S cones through the dorsal lateral geniculate nucleus of the thalamus to the primary visual cortex V1, traversing the associative visual areas V2, V3, and areas like the fusiform gyrus, the lingual gyrus, and the V4 area, to the angular gyrus implicated in higher order processing of colors along the ventral stream through the posterior inferior temporal cortex, central inferior temporal cortex, and anterior inferior temporal cortex in the left hemisphere. The color processing is accomplished between two color spaces by wavelength sensitive cortical neurons with axons in the cortical subcortical circuits. The female model begins with the retinal S cones and goes through frequency sensitive neurons of the dorsal lateral geniculate nucleus of the thalamus, with axons ascending within the dorsal stream to the primary visual cortex V1 and empty region in the right hemisphere. We estimated the origin of the C peak and the S peak. The C peak occurred at the second harmonic at about an arterial length of 14 millimeters and at a frequency of 17.2, which would correspond to the occipitotemporal junction in the mouse brain. The S peak occurred in the third harmonic from about an arterial length of 4.6 millimeters at a frequency of 25.8 Hz, which will correspond to the ganglionic branches in the mouse brain. In conclusion, our findings clearly demonstrate that the C peak corresponds to 17 Hz, which is the beta band synchronization in the left cortex in male mice, while the S peak in the female mice corresponds to a higher frequency range, which correlates with 25 Hz gamma band synchronization in the right visual cortex. The findings demonstrate clearly that Fourier time series analysis as a computational method could be applied to the study of the visual cortex. Thank you very much for listening.